A lot and hello everybody. Thanks a lot also for inviting me to this highly interesting webinar on the example of our cannabinoid receptor 2 agonist program. I would like to give you a holistic overview on a small molecule derived drug discovery program. And as you can guess by all these logos here on the title slide, a huge collaborative efforts between industry and academia stands behind this talk. And therefore, I would like also to start off by acknowledging all involved colleagues for the great contributions and the fantastic collaborative spirit. On the left, you have lots of colleagues from Roche coming from different disciplines, and on the right, many academic colleagues, many of those being KOLs in the field of endocannabinoid research. So what am I going to talk about today? First of all, I would like to explain to you why Hoffmann-La Roche is interested in the CB2 receptor and what challenges we do see on the way towards clinics. On some of them, I will allude a bit more in detail, such as the expression and detection of the receptor. Then I will show you how we arrived at novel ligands and give you a first glance of their in-depth profile. And later on, I will draw your attention to some of the chemical probe work which we have done. Let's start with a few words on the endocannabinoid system, which is a very important lipid signaling network, which is conserved through all vertebrates and plays a major role in many health and disease conditions. The key signaling molecules are these two endocannabinoids, 2-arachidonoglycerol, 2-HE, and anandamide, AEA. They activate the G protein-coupled receptors, cannabinoid receptor type 1 and cannabinoid receptor type 2. Furthermore, many anabolic and catabolic enzymes belong to the endocannabinoid system, for example, fatty acid amide hydrolase FAR. So why are we particularly interested in the CB2 receptor? It is because it plays a key role in inflammatory processes and is mainly expressed on immune cells. Upon or as a consequence of an inflammatory stimulus, leukocytes adhere to the endothelial cells, and this triggers a lot of inflammatory and tissue damaging events. And upon the activation of the CB2 receptor, you can block this leukostasis, reduce chemotaxis and oxidative stress, which at the end of the day prevents tissue from being damaged. Such tissue damage, which could come from ischemia perfusion, chemical or mechanical injury, and fibrosis, can lead to the development of both acute and chronic kidney diseases. And since there's a high unmet medical need for both groups of indications, we embarked on a CB2 agonist program, trying to come up with best in class ligands and to evaluate their potential for the treatment of kidney diseases. This graph here shows you research and development phases for a typical small molecule drug discovery program, starting from the initial idea up to launch. For each of these phases, there are project-specific challenges, and I would like to mention some of the questions we asked ourselves when we started the program. There were, for example, severe doubts regarding the specificity of available antibodies, and hence we asked the questions, where exactly and under which circumstances is CB2 expressed? What happens after the binding event? Can we come up with good chemical starting points? And latest in the lead optimization phase, you need to be able to control selectivity against anti-targets and have an eye on species differences. Is there a good in vivo efficacy model around which allows for a go, no-go decision? Can you come up with an exposure response relationship to inform dosing for clinical trials? And also very important, is the principle and are your compounds safe and developable? Very important for the translation is, will the animal data translate basically to the human situation? So will they be efficacious for the human situation? Will you see efficacy in human trials as well? That's where most drug candidates fail at the end of the day. And also, what is also very important, especially for lengthy clinical trials, is can you come up with a biomarker? I will talk you through some of these aspects now, and we'll start with the CB2 detection question and end with the biomarker question. As mentioned, specificity of available antibodies seems to be insufficient. And when we started our program, we got hold on the commercially available ones and tested them for a couple of cells which are potentially positive for CB2. Here you see a representative Western blot using a Santa Cruz antibody. To support the sample characterization, we did N-linked deglycosylation to get a specific band shift, but still the picture does not look very good. On the search for an endogenously expressing cell system for screening purposes, we tested lots of cells, but found CB2 protein only in a few of them, such as, for example, in Trocat cells. And we asked ourselves, why is this the case? Is it due to the limitations of the available tools, or is it because these cells do not express CB2? Therefore, we went one step back and asked the question, which cells can express the CB2 receptor? And we did an RNA-seq analysis of around 500 human cancer cell lines and arrived at this picture here, which tells us that only few cell lines do express high amounts of CNR2 message, while also only few cell lines do not express the receptor at all. In order to get a signal in the Western, we needed to have at least 4 RPKM. For example, we got a Western signal for Trocat cells. The overall expression rank order for CB2 is that B cells are the highest expressors, followed by monocytes and macrophages, and then by T cells. And these data are paralleled by data from the ImGen database, where researchers looked at the messenger RNA expression in mouse immune cells. For mice, however, there's no antibody available at all. And therefore, we came up with this highly specific radioligand, which carries 3 tritium atoms at this position. It is a 2 nanomolar binder for human CB2 and 3 nanomolar for mouse CB2. It is not only highly selective against the CB1 receptor, but also against more than 80 common off-targets, including key proteins of the endocannabinoid system. With a log-D of 3.3, the molecule is way more polar than reported cannabinoid-derived radioligands, and therefore could be successfully applied in tissue binding studies. Here you see that you get a highly specific signal in CB2 positive spleen tissue of wild type mice, while there's no signal at all for 2 different strains of CB2 knockout mice. Consequently, we could use this radioligand for the detection of CB2 in mice and also in other species. What about the quality of the available tool compounds? When we started our activities, we got hold on many of the reported tool compounds for validation purposes to set up our in vitro and in vivo assays. Quickly, we found that they were suboptimally characterized, and therefore we teamed up with a couple of KOLs to do a thorough profiling exercise, which led to a couple of interesting findings. For example, we found that some of the ligands are selective against the CB1 receptor only for the human situation, but not for mice. And as a consequence, we had to be very cautious with regards to the interpretation of reported mouse in vivo efficacy data, especially since CB1 and CB2 activation often leads to counteracting effects. At the end, we reached consensus that HU910, HU308, and JWH123 are the best suited CB2 ligands for studying the role of the receptor in biological and disease processes. In parallel to that, we started to come up with new chemical matter. And among all these options we generally consider to do so, we have chosen a high throughput screening campaign and an information-driven approach, which was based on a previous in-house CB1 inverse agonist program. Later on, we also got access to HU910 from the Meshulam lab, which is a cannabinoid type ligand. On the example of the triazolopyrimidines, which are HDS derived, I would like to show you now some snapshots of the hit to lead process. Focus will be on early ADME properties as well as on the selectivity against CB1 receptor, since CB1 activation, especially in the brain, and its associated psychotropic side effects were considered to be a showstopper for chronic kidney diseases. Screening around 900,000 molecules provided us with this highly attractive starting point, which is a 1 nanomolar full agonist for human and mouse CB2. Functional selectivity against CB1 is 2,200-fold. CB2 binding affinity, or Ki, for a human receptor is 29 nanomolar. Calculated lipophilicity is a bit on the high end. The molecule permeates well through membranes. Solubility is poor, and also microsomal clearance needed to be optimized. The molecule offers 3 exit vectors for modifications, and the metabolite identification studies indicated that this morpholine ring here is most vulnerable. Therefore, we started our optimization work with position 5, followed with positions 1 and 7, looked also into core modifications, and at the end, tried to combine most favorable residues. I've cherry-picked some representative examples. All of them are full agonists on both species, and all of them and the data are color-coded, while green stands for favorable, orange signalizes there's room for improvement. Morpholine replacements led to a drop in functional selectivity. We screened here different ring sizes and found 500 rings to be best suited, leading to first picomolar inhibitors, or agonists in this case. Introducing 2 fluorine atoms here improves the microsomal stability. When you bring in here a hydroxymethylene group, you bring back the functional selectivity, and when you combine this with a second hydroxy groups in this 3 position, you completely abolish the interaction with the CB1 receptor. Also, aliphatic heterocycles are well tolerated in position 1. However, clearance remains high. With this bis-chlorobenzyl derivative, we arrived at first picomolar binders. C log P, however, is quite high. With this methyl sulfone residue, C log P looks already much more friendly. And when combined with a 3-hydroxypyridine in position 5, you end up with a highly potent and highly selective molecule with decent overall properties. The same can be achieved with such pyridine-derived or such tetrasol-derived ligands. With regards to position 7, none of the investigated residues was superior to tertiary butyl, so we simply kept that. Also, 2 substituted trisolopyrimidines are highly potent and selective CB2 agonists. However, their SAR is a bit steeper. When you try to bring a lead into, or convert a lead into a drug candidate, some of these angry-looking guys might get in your way. Their names are, for example, bioavailability or drug-drug interaction. In the next slide, I will show you examples from compounds representing 3 chemotypes which made it to the other side. All of them are highly potent CB2 agonists across species. Some exhibit even picomolar EC50 values. Functional selectivity is minimum 900-fold. Compounds have a reasonable lipophilicity. They permeate well through membranes, and also most solubility data are favorable. Clearance in human microsomes and hepatocytes is low, and mouse pharmacokinetic data suggests that the molecules are well suited for being administered in mouse models of kidney disease orally. Furthermore, free fractions up to 13% could be achieved. Here you see the concentration time profile of these 2 ligands. 1 hour after an injection, IV injection of 0.5 mg/kg. In both cases, the free drug concentrations are several times above the mouse EC50 values. Here, the PK profile of this pyridine. Again, in mice, the blue PO curve tells us here that the molecule is fastly absorbed and reaches high maximum concentration. In its bioavailability is 41%. With regard to early safety assays, none of the molecules does significantly inhibit the three most important cytochrome P450 isoenzymes, nor the hERG channel. And taken together, this means that the molecules were well suited for being administered in vivo efficacy models. Here you see three of the ligands again on top. In addition, this previously introduced pyrrosol and AG910. All of them are highly potent and selective CB2 agonists. Nevertheless, they span a very broad range of physicochemical properties. But despite of their diversity, they do bind. Here you see the 3D shapes of the molecules. They do bind to the same binding cavity as illustrated by an overlay of the pharmacophores within the agonist binding pocket. Here also in a space-filling illustration. But even more important, they also do the same things in vivo models of kidney disease. But before talking about that, let's introduce the pathology of diabetic nephropathy quickly. As mentioned, available treatments for acute and chronic kidney diseases are suboptimal, and diabetic nephropathy belongs to the group of chronic kidney diseases. Diabetes and acute kidney injury are among the main drivers for the development of diabetic nephropathy. Inflammatory processes are followed by a cascade of events leading to fibrosis, albuminuria, and at the end, renal failure. The in vivo data we generated cover the following elements: ischemia reperfusion, inflammation, glucostasis, fibrosis, and albuminuria. We first investigated our ligands in an acute kidney injury model. In this model, you block the blood flow to both kidneys for 25 minutes. You administer the CB2 agonist via garage 30 minutes prior to the surgery. And after 24 hours, you analyze the blood for efficacy, for the efficacy parameters, creatinine and urea, as well as for the biomarkers NGAL, KIM1, and osteopontin. These four molecules were investigated at a dose of 3 and 10 mg per kg PO, and all of them significantly reduced plasma creatinine as well as plasma urea levels. What about the biomarkers? All molecules also reduced NGAL, a marker of kidney damage, and for KIM1, kidney injury molecule 1, a marker of tubular injury. There was a clear trend after 24 hours and significance after 48 hours. Osteopontin, a player in wound healing, was reduced by these two molecules here down to the sham values. So overall, all four molecules improved also the biomarker values, and this is something which goes beyond what normal reference drugs do, and prompted us to go one step further into a subchronic model, the so-called unilateral ureter obstruction rat model, which covers the fibrosis component of chronic kidney disease. Here you ligate one of the two ureters, thereby you block the urine transport to the bladder, causing a back pressure here, which leads to the development of fibrosis. A picture of the obstructed versus the contralateral kidney. The CB2 agonist is administered 1 day before the obstruction via food admix, and after 8 days of treatment, the histological evaluation of the kidney follows. The readout is collagen 1, which is proportional to the degree of fibrosis. These 5 diverse, highly selective and potent CB2 agonists were investigated at a dose of 3 mg per kg via food admix. Compound number 6 is enalapril, an ACE inhibitor, which is used in the clinics for the treatment of chronic kidney disease. Let's start with the triisolopyrimidine. On the left, you have the collagen 1 levels for the obstructed kidney. On the right, the respective values for the contralateral kidney. The difference between both gives you the window which is accessible for pharmacological intervention. And after 8 days, the molecule showed a 40% improvement on fibrosis. We followed up with this pyrosine and this pyrosol and could confirm the effect. Here also enalapril at a dose of 32 mg per kg was included. Also, this pyridine here showed a significant improvement of fibrosis, and together with our partners from the Hebrew University, we also investigated HU910, cannabinoid-inspired ligand, and could again show strong antifibrotic effects, which were even maintained when the drug was administered several days after the obstruction. And that prompted us to go also into a chronic kidney disease model with this molecule. In the lab of Professor Remuzzi, 10-week-old BTBR-OB mice were treated with a daily dose of 10 mg per kg HU910 for 11 weeks. At different time points, systemic blood pressure and glucose values were monitored. And after total 21 weeks, significant improvements of kidney function, such as a reduction in albuminuria, as well as an improvement of the inflammatory state, for example, as shown here by a reduced macrophage infiltration was observed. Importantly, these effects were independent of antiglycemic and hemodynamic effects. We went one step back and asked the question whether we could also show that CB2 activation reduces the number of leukocytes which adhere to endothelial cells. And this was indeed possible by going for a uveitis model. Uveitis is an inflammatory front of the eye disease, and the setup is depicted here. You anesthetize the animals, then you inject intravitreally lipopolysaccharide S to trigger the inflammation. 10 minutes later, you administer a CB2 agonist via eye drops. A couple of hours later, you inject a fluorescent dye which labels the leukocytes, and then you follow the fate of these leukocytes via intravital microscopy. Picture we arrived at is depicted here. You see upon treatment with a CB2 agonist, you could significantly reduce the number of adherent leukocytes per square endothelium as compared to vehicle. The bars of this chart were derived from movies of this type. This is from the iridial microvasculature. On the left, you have LPS only treated animals. On the right, LPS plus CB2 agonist treated animals. Clearly, you can see that much less of these white dots here, which are leukocytes, adhere to the blood vessels in the treatment group. And with this extra in vivo efficacy data, we felt very confident that CB2 activation has high potential for the treatment of acute and chronic kidney diseases, assuming that these rodent data would translate towards the human situation. Still, we didn't have a biomarker in hands at that time. Why is this important? It's important because we talk here about lengthy and costly clinical trials, and we would like to have a sort of an interim readout which tells you whether you have a chance to meet a hard clinical endpoint or not. And that's also the goal of such a biomarker, to establish a correlation between CB2 activation, a CB2-mediated readout, and the clinical endpoint. And there are different options to do so. Either you go for direct downstream effectors, which prove to be difficult for CB2, or you go for a small molecule derived probe, such as CB2
development process.
Or you go for a small molecule, or you go for positron emission tomography probes, PET probes. Such probes need to fulfill a couple of criteria. They need to be as potent as possible on target and selective against anti-targets. Furthermore, they need to carry a reporter group, such as in our case, a fluorescent probe, for example. Ideally, they come along with an inactive control compound, which is chemically closely related. For our probe work, we use the so-called reverse design approach, which means that we started from molecules which have been optimized by medicinal chemists. Here you see 6 molecules which we used, representing 5 chemotypes. All of them are highly potent CB2 agonists, except for that one, which is a potent inverse agonist. The ligands are not only selective against CB1, but also against the panel of around 80 representative off targets. And for our probe work, we embarked on a couple of collaborations with academic partners. Here you see a table which gives you a snapshot of still ongoing work. This molecule is the tritiated ligand I introduced before, which was used for binding studies and radiography studies. Here, tritiated inverse agonist, which was used for kinetic studies. This is the same molecule again, but now with a carbon-11 reporter group for in vivo PET studies. Here you see a molecule carrying two different fluorinated moieties, which was used for structure determination of the receptor via NMR techniques. Here an alkyne labeled molecule, which can be applied for CB2 detection using stimulated Raman scattering. In the light of the limitations of the existing antibodies, especially fluorescently labeled molecules can be highly versatile tool, and were investigated here an early example carrying a nitrobenzofurazane reporter unit. Also, covalent ligands such as this fluorosulfonyl derivative are highly versatile tools, for example, to support receptor crystallization work, or they're also an entry ticket for all sorts of bifunctional probes, such as this molecule here, which carries a photoactivatable diacerine moiety for photo cross-linking to the receptor, and an alkyne handle, which can be used to click all sorts of tags onto it. All that work has helped tremendously to better understand CB2 expression and mechanism of action, and ultimately provided us with small molecule derived biomarker options. In the last 1 or 2 slides, I will show you a few applications of our fluorescence probe work, which was intended to detect CB2 in cells and to provide us with a surrogate marker of target engagement. First generation probes where we placed the fluorophore within the binding pocket suffered either from insufficient in vitro potency or specificity in fax. So we had to change our strategy, and in a second approach went for molecules consisting of a CB2 recognition element, a linker, and a fluorescence tag, with the aim of placing the fluorescence tag towards the extracellular space of the receptor, where it cannot interfere with the binding of the recognition element anymore. And this approach was successful for 2 different chemotypes, which I will show you now. Dimitri and David used them, for example, for homogeneous time-resolved fluorescence measurement to determine equilibrium and kinetic binding data. This technique combines normal FRET technology with time-resolved fluorescence measurements and allows for a removal of short-lived background fluorescence. From the FRET signal, you can get to equilibrium tracer binding data. The first probe which was used is this DY480XL labeled molecule, which serves as a FRET acceptor, while the donor are engineered hex cells overexpressing CP2 and carrying a terbium cryptake. Here you see the HTRF emission versus probe concentration, green line specific. And if you keep the probe concentration constant, here we used 150 nanomolar, and then come in with increasing amounts of cold ligands, you get such sigmoidal curves, which you can convert into Ki values using the Chern-Krusov equation. And importantly, these fluorescence-derived Ki values match pretty well with radioligand binding derived data. Another application is shown here. It's an example from the Nazare group. Here, an Alexa Fluor 488 labeled molecule was used to detect CB2 positive cell populations via fax. We used here CHO cells overexpressing CB2, human CB2, mouse CB2, or human CB1. And you can see from this graph that you get a highly specific signal for CB2 in blue, while there's almost no interaction with the CB1 receptor. Importantly, also, the blockage of the signal with cold ligands was successful. Here, silicon rhodamine labeled a close analog of this molecule, which was used for lifestyle imaging studies. CHO cell lines were co-stained with the reddish fluorescent dye and the cyan Höchst nucleus stain. In CB1 overexpressing cell lines, there was almost no labeling, while in CB2 overexpressing cell lines, the dye readily incorporated into the cell membrane and after 10 minutes reached almost all internal membrane fractions. Also here, blockade with cold ligands was successful, and even more important, we could also show that it works for endogenously CB2 expressing cell lines such as mouse splenocytes and human macrophages. And very recently, we could also show that the molecule works also in vivo in zebrafish embryos. After an IV administration, the ligand freely circulates in the blood vasculature and is then, after a while, extensively taken up in the caudal hematopoietic tissue where the immune cells form. It co-localizes the reddish dye, co-localizes with the local residual macrophages in cyan before they are removed from the blood circulation. Here you see that also in a movie looking with two different channels at the same tissue basically. Let me please sum up. I hope I could convince you that significant progress was made toward bringing a CB2 medicine to bedside. A lot of progress was made with regards to CB2 detection and expression, regarding mechanism of action, and by signaling there's still a lot to do, but now we do have tools in hand to study that. With regards to chemical starting points, CB2 is clearly a tractable target. Selectivity against anti-targets and species differences can be controlled, safe and developable assets are in place. In vivo efficacy data mirroring several aspects of acute and chronic kidney diseases has been demonstrated in rodents, and therefore CB2 activation holds great potential for the treatment of human disease, for acute and chronic kidney disease in humans, assuming that these data will translate towards the clinics. Also, with regards to small molecule-derived biomarkers, a lot of progress was made. Many of these achievements were only possible through fantastic collaborations with academic partners, which were not only mutually rewarding and of a highly cross-fertilizing nature, but also a lot of fun. With this, I'm at the end. Thank you for your kind attention and would be happy to take any question.
Thank you for your wonderful presentation. The floor is open for the question. Anyone have questions?
Good afternoon.
Hello.
Dr. Andres, you can ask your question.
Good afternoon.
Yeah, we can hear you. Dr. Grether, can you hear it? Dr. Grether, can you hear?
Yes, I can hear you.
Yeah. Dr. Andres, you can ask your question now.
Excuse me,
Yeah, Dr. Andres is having some questions.
but I didn't understand the question so far, unfortunately. Dr. Andres, would you please repeat the question?
I'm getting one more question from Diva. Diva, you can ask your question.
Yes, hello. Thank you for your presentation. I wanted to ask, when you were talking about the lead optimization, sorry, what were the methodologies and the tools that were used, if that's information that can be shared?
Well, for the lead optimization, you use everything which fits to the program. For example, if you have 3D structures, which we don't have, or which we have now, but we didn't have for CB2, then you can use structure-based drug design, which was not possible at our time for CB2. So we just used an optimization of where we explored the different regions of the molecule, which has a central core and 3 exit vectors. And what you do then is just to replace the different elements by other residues. And you do that in a way that you not only look into maintaining or further improving the in vitro activity of the ligands, but also you need to keep an eye on the overall properties of the molecules, physicochemical properties, metabolic stability, drug-drug interaction, and so on, that you make these molecules not only active for the receptor, selective against anti-targets, and then also having in mind what a drug, what sort of drug properties the molecule should have at the end, and design and synthesize in this direction. Does this answer your question?
Yes. Also, I just wanted to make sure, was there any sort of like AI or computational methods used?
We used, this was before the AI hype. What we do, of course, is we now regularly apply machine learning tools to streamline our design. We use design hubs to rank our ideas, and but always with cross-checking the AI proposed with experimental data. So this is very important if you do drug design, that especially when you're tapping in a new area, that you always cross-check the proposals, you synthesize the molecules, and then you experimentally measure and validate the predictions which have been made by the AI tools, and continuously improve these tools. And this is how you do nowadays the drug discovery in the big pharma companies. And this helps you basically get forward faster than previously. So it accelerates the drug discovery. So there's a coexistence of measurements and AI.
Thank you for your